Okay, so for DNA sequencing, you have a short sequence of DNA that you want to then find out what that sequence is. Okay, you put this into a little tube. In there then you'll put a whole set of other nucleotides. So there'll be a mixture of adenine, thymine, cytosine and guanine. You'll put a primer, which is a short sequence of nucleotides which is complementary to the sequence that you are trying to find out about. You'll put a buffer in there. The buffer then is to maintain the pH and make sure that the enzyme, the sequencing enzyme, which is DNA Taq polymerase, will work. And then you'll have special nucleotides, and these are nucleotides that will stop the sequence. Okay, so they often get called DDNTPs. Okay, dideoxy, and then there'll be an adenine one, a thymine one, a cytosine one, and a guanine one. So it might be DDATP, DDCTP, DDTTP, DDGTP. These then stop the sequence, and that's what we're looking at today. So, on the activity I set you out, question number 1 says write down a single-stranded DNA sequence consisting of 27 deoxyribonucleotides. And it says do it in the 3' to 5'. Don't worry about that too much. So I've written a sequence of 27 nucleotides. So remember that's C, G, A and T. A random order of them. Okay, and then I've labelled it 3' to 5'. So that's question number 1 done. Question number 2 then says write down a primer. So simply keep it to 5 nucleotides. Now it says this time it's 5' to 3'. So it's got to be the other way around. So if this is my 3', this has got to be my 5', so it's got to be complementary. So we know that cytosine bonds to guanine in base pairing. So I get a C to a G, and then my G gets a C, my A gets a T, my T gets an A, and my C gets a G. It's 5 bases long. Okay, so these 5 bases then, okay, are always going to anneal to the first 5 bases of my DNA strand. So I am now interested in the other 22 bases. Now obviously we've written these down, but in reality we wouldn't really know what they were, and we'll be sequencing to find them out. So it says, below are different reaction tubes. Your task is to write down all the possible complementary strand lengths that terminate with the D-deoxy analogue present. Okay, so we notice then the first one I've put here, I've got A, T, G and C. So A, the analogue is DDATP. Okay, and it's going to stop the sequence any time that A would be put in place. Okay, so the first thing I'm going to do is I'm going to write down my primer sequence, my G, C, T, A, G. There's my primer. I'm going to just put that in a line. So from here then we're going to put down the complementary strands, and any time this A gets put in, we stop it at that spot. So we're here. So opposite a G comes a C, opposite an A comes a T, and opposite a T comes an A. Okay, and I'm going to stop it there because this has come into place. I'm going to put a little star. And so I've got a sequence now of 1, 2, 3, and I'm just going to write 3 here. So then I'm going to do the same for the next strand, C, T, A, G. There's my primer. So I'm going to go here, that's a C, that's a T, that's an A because it's already gone into place, and now we come to the next A, we go there, and that's got a sequence of 4. And I'm going to do that continually until I've done the whole length. Okay, sorry, I was just putting on my computer. Okay, I'm going to keep on going. So opposite this A then comes a T, opposite this G comes a C, opposite this C comes a G, G, C. Now I've got my next A coming in. So I'm going to count this, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm going to put a 10 there. So I'll keep doing that for all of them for this sequence. Then I'm going to move on to my next one. Okay, so this is DD DTP. So I do the same again. I'm going to put my primer in, GTA, like that, and put it in line. So opposite my G here, opposite my A, I put a T. But because I put this T in place, I put a star and I put a 2. And I keep on going then until I've gone the whole length of my primer. So I go C, T, and then an A opposite that one, an A opposite that one. I've got a T here, so that becomes a T down there, and I put a star. I count it 1, 2, 3, 4, 5. I'm going to put a 5 there. And then I'll continue writing these primers all the way down here for the whole length of this one here. Then I'll move on to my next one, which is DDGTP. Okay, so I put my primer down again, put it in brackets. So opposite as G goes a C, so straight away that C go, oh, I've made a mistake, sorry. So yeah, and then I put my T in, opposite an T goes an A, opposite a T goes an A, opposite an A goes a T, opposite a G goes a C, opposite a C, here we go, puts my G in place. Count them, 1, 2, 3, 4, 5, 6, 7, I put a 7 there. So then I do the same. Okay, and I copy it out, A, T, C, there's that first G, there comes my second G, now I've got 8. And then I do exactly the same again, all the length along my chain. Okay, so any time that the G would go in, I put a star next to it and I just count the same as I would have done with the T's and the A's. Then I move on to the C's. I put the primer down first. So this is DDCTP. Put the primer in. So here we go then. I've got a G first. Opposite a G is a C. This C would have been put in. It would have stopped the sequence dead. And I just say I've counted 1. Okay, and then I do the same again until I get to the next time a C would be put in a complementary place. So I go C, T, A, T, C, put a star there. 1, 2, 3, 4, 5, 6, and I put a 6 there. And I keep on going then all the way along and all the way along my length. Okay, so I have all the C's at the very end. Okay, so I've just done a couple for each one, but you would go through the whole length of your sequence for each primer. Okay, and you stop them when it gets to the complementary base. It says then, question number 4, count the number of nucleotides in each fragment you have synthesized and write them down next to the fragment. So we've done that as we've gone along. 3, 4, 10, 2, 5, 7, 8, 1, 6. Okay, so some of these then are going to get up into the 20s. And remember, the first 5 are the primer, so we're interested in the other 22. Okay, so you should have numbers then that add up to 22. On the final page then of the worksheet, what you now do is you write these out. You write these out in order, one under the other. Okay, so if we write the primer at the top. Okay, so there's my primer. I've just put my line all the way down. I'm going to look over. So the shortest one is a C. So I'm going to write C. The next one then is CT because it's got 2. I'm going to write that one under there. I'm going to find which one. So CTA has got 3. So I'm going to write that one. CTA. I've got 1, 2, 3. I'm going to find which one has got 4. CTAA has got 4. Okay, and you're going to do it all the way down then. Okay, until you get to 22. And 22 plus a 5 makes you 27. And this then should be the complementary sequence to what was originally put in your tube. And so it should tell you what the sequence is. Right, I am hoping now that helps you a little bit. Okay, if you're struggling, feel free to get in contact. But this should explain how to do the activity I have set you. Okay, brilliant.